All right, so are you ready for a little bit of theory? Yeah, okay. Variant calling with HaplotypeCaller. How does it work? So we're in this part of the pipeline. I just showed you, you do the per sample calling with HaplotypeCaller and then the joint calling across the GVCFs. We're going to focus in on what that does. Now, HaplotypeCaller is a tool that is designed to identify germline, SNPs, and indels with very high sensitivity because we don't want to miss anything that might be real in our sample. And now, traditionally, what you would do is you look at, let's say you're looking at a particular position in the genome, you look at how the reads are aligned, and you just see if there are mismatches to the reference, how many are there, if there's about all the reads or about half the reads, we'll say, okay, that's a likely SNP at this site. That's the basic idea. Now, the problem is that data is messy, and in a lot of cases, we don't have something as clear-cut as this, especially when we throw in indels, which are insertions and deletions of short amount of sequence. It can get pretty messy, and we need more sophisticated algorithms to deal with that. So the way HaplotypeCaller is going to work is it's going to do a lot of work under the hood when it's looking at any given region of the sequence. And so the first thing we want to do, just to be economical, is we're not going to do all the work on all the sequence because there are large areas of sequence where it's obvious that there are no variants, where there's only a tiny sprinkling of things that are clearly errors. Things like if you have one base out of a big, what we call a pileup, when you have a lot of reads piled up in a particular region, it's called a pileup. There's one base that's different for the reference. It's very unlikely that this is real. This is most likely an artifact. And if you have a wide area of the sequence where you only have things like that, you don't want to waste any time making any complicated computations. You're going to want to focus on areas where there is some evidence that there might be something real. It can be a few mismatches. It can be evidence of insertion or deletion, or something called soft clips. And when we see that, we're going to say, okay, there's something likely that we want to do here. And so that part of the process is called identifying active regions. And that allows us to focus our energy on those regions and then try to dig more to see what's happening there. Once we have an active region, we're going to do a reassembly. We're going to take the reads, create an assembly graph, and based on that, we're going to derive what are the possible haplotypes, the possible ways to reassemble the sequence in this area. Once we have that, we'll have several alternatives for what the sequence could be in our sample in this area. We're going to do some computation to decide which one or which ones, depending on ploidy, are the most likely to be biologically what belongs to our sample. And then finally, we're going to run some final computation to actually derive a genotype and decide for a particular site, I have AC SNP, and I decide that this sample is heterozygous, that sample is homozygous variant, things like that. So that's a genotyping step. Those are the four main steps of how HaplotypeCaller works. And now we'll walk you through a little bit more detail about each step. And again, please feel free to interrupt me at any time if you have questions. All right, so I just walked through that. Identifying active regions, how does that work? We're going to take a sliding window, meaning say we're looking at a certain length of sequence. Say it's 200 base pairs, 500 base pairs. I forget the exact setting by default. But you're looking at a certain length of sequence, and kind of as you go, recording how many events you have within that length. We're counting how many mismatches we have, how many indels, and how many soft clips. Does everybody remember what soft clips are? Does anybody know? Soft clips? Yes, no? Soft clips are when you have the aligner, the mapper that aligns the reads to the reference, is aligning your reads, and then at some point decides, you know, I don't know how to reattach this sequence back to the reference. This doesn't make sense. This is too different from the sequence. I'm just going to say after this point, just ignore everything else. Just ignore the rest of the sequence because it's not making sense to me. Soft clips are really interesting, and we'll see that in the practical section. So we're counting them. We're counting how many of them are within our active region. And we are taking these counts as a measure of entropy. Entropy is just, you know, the amount of chaos we have, the amount of things that are not orderly and neatly matching. And we can kind of take that as essentially a function. We apply some smoothing, and on top of that, we kind of derive, we measure, it's basically a local optimization problem where we look to see if we have any local peaks. And if we hit, for any particular region, if we get to a point where we cross a threshold and there's enough happening, there's enough things that we're counting within that area, we would say this is an active region that is worth investigating in more detail. Go ahead. Oh, talk louder? Okay. Sure. All right. Got everything so far? Or should I repeat? No, okay. All right, so we declare an active region. We say this is within this window. We want to figure out what's happening. That's great. So how do we figure that out? The really key thing about HaplotypeCaller and how it works is that once it decides it has an active region, for that active region, it is going to throw away all of the mapping information for all the reads within that. So all the hard work that PWA did to align those reads, it's throwing it out the window. Now, it's not entirely true that it's completely disregarding PWA's decision, since it did tell us where the reads and roughly what region the read belongs. And that's what BWA is really good at, and mappers in general, are really good at telling you roughly where a read belongs. But they're not so good at giving you a very detailed alignment of the read to the reference. And part of the reason is that the way they're scoring some words will penalize gap, sometimes unfairly. So it's basically, if I have to insert a mismatch into the alignment, it's giving me a penalty. If I have to insert a gap in the alignment, it's giving me a bigger penalty. And the problem is when you have a single reference and you have a large insertion, the gap penalties are going to add up to the point that the mapper is going to say, forget about it. This is not aligning, and that's where I'm putting in a soft clip. And that's your problem. Because beyond a certain length, which is variable depending on factors, you're not going to be able to identify those indels very precisely. In some cases, you might not see them at all. And so the mapping information that BWA gives us is great, but the alignment information not so great, because it's doing things only read per read. Whereas now what we have is a whole bunch of reads. And what we can do is put them through a graph alignment process, build a graph of those reads. And by building that graph, we can reconstruct what are the possible ways that these reads could explain, reconstruct the sequence that we actually have in the sample. And we can also, the nice thing is we can reconstruct the haplotypes, and that's why it's called the HaplotypeCaller, the possible haplotypes that we have in our sample. So we generate that with an assembly graph. And the idea of the assembly graph is really to take each read in a row and add it to the others and see what are the divergent paths that are in my graph. And the haplotypes just derived from looking at all the possible paths through the graph. That's basically how it works. And that gives us a lot more power to identify insertions and deletions, since now we're not constrained to score things directly relative to the reference. We're just looking at how everything fits together, which is very powerful. The nice thing about doing this graph assembly is basically what I've been explaining, that you can recover indels at a much higher rate, especially large indels, especially in areas where the indels are mostly covered by ends of reads for whatever reason. And so this is showing you an alignment, a read alignment by BWA before assembly process, and then after having reassembled with HaplotypeCaller. And you can see that before I had a few reads where BWA had identified there may be something happening here, deletion. But afterwards, I have much better support, and I can call this variant with much higher confidence. Okay. And we'll go through an exercise of that in practice after the break. Yeah.
HaplotypeCaller will out another body of verify.
It is possible to make it do so. It does not do so by default. By default, it doesn't create a whole new BAM file. But you can ask it to create what we call an output BAM, which will output the sequence, the reassembled sequence for the active regions. So it won't give you an entire BAM, and if you ask it, and ask for practical reasons, that's difficult to do, and it takes up a lot of space. But you can have slices of the active regions reconstructed, and you'll see that in the practicals. Mm-hmm. Yeah, yeah. And for example, in the Nomad viewer, when you're looking at variant calls that are made in Nomad, you have the option to see the output BAM file from the original data.
So these haplotype calling is something we should do regardless if you're looking for variants, since these are variants, and why BWA wasn't extended to produce such
It's a lot of extra work, because the nice thing about BWA processing reads independently is you can parallelize and you can multi-thread quite extensively. And BWA is already doing a lot of work. But this requires looking per section of the genome and actually doing all that reassembly. It is a lot of extra work. And when we originally came out with this this tool, it took much longer to get variant calls compared to traditional high-level callers. People were actually slow to pick it up, to start using the tools, in part because it took so much longer to run. Now the situation has massively improved, in part because, well, our developers made the tool faster and found some optimizations and so on, partly because some external tools companies produced some accelerated versions that run quite a lot faster, like using various software engineering tricks. But it's a lot of extra work.
Is this like I should do for any kind of work I'm doing? Regardless I'm doing variant analysis, should be done.
I would say it depends what you're doing. Because, for example, if you're looking at copy number variants, most of the time you don't need to have super fine alignments, as long as you know the read belongs in this area. You can count it as part of the coverage, and that's all you want to know. So you don't really need to have super fine alignment for that. It's mostly for short variant calls that you need very precise alignments, and to some extent for structural variation, if you want to identify particular breakpoints, there you have to have more fidelity in the alignments as well. But so this is something we do for both Germline and somatic short variants. And you'll see tomorrow morning that Detect 2, which is the somatic version of HaplotypeCaller, does this, uses the same code under the hood to do the exact same thing. Yeah?
So is this algorithm exactly the same as in the old Rala?
The comparison to indel realign is useful, but this is a much more sophisticated version of what indel realigner did. So indel realigner was an old tool that was a pre-processing tool that didn't solve the same problem, but it did so with a much more naive approach. It didn't use a graph assembly. It did a much simpler multiple sequence alignments. This is a lot more powerful using the graph because it's much less constraints in terms of what it can come up with. That makes sense. Any other questions? Like that is the most important part of how HaplotypeCaller operates, really. One of the nice benefits of doing this is that if you have, sometimes you have some slight complexity in the sequence. For example, if in your reads you have two bases, you have, it's basically, you can interpret that as both an insertion and a mismatch. But you can get it as an insertion and then a mismatch, or a mismatch and then an insertion. And with a traditional HaplotypeCaller, because the mapper would typically kind of randomly produce both possible alignments, since it's doing the reads independently, you would end up with a mix of both, and then in the variant calls, you would have a whole mess right here. You would know there's some kind of variance, but it's inconsistent how it's represented. With the graph assembly process, you will come up with this, understand that it's a T turning into AC, and the caller will emit one representation. Which one it chooses is not necessarily easy to predict. It depends on the data, but you will have the same representation throughout, and you'll have a consistent, like a clear variant call coming out. So that's nice. Another nice benefit is that you get physical phasing. So physical phasing is when you have variants that are located on the same or the other molecule of DNA. Like physically, are they together on the same molecule of DNA or not when they're close together? So are they in cis, same molecule, or trans, the other molecule? If we're looking at the diploid case. If you have a hydroploidy, it still applies. But basically, HaplotypeCaller is going to tell you if your variants are on the same haplotype or the other haplotype. Okay, so that's it. And there's some biological implications and some implications of the, in terms of the interpretation of the effects of the variants. Not going to get into it, but basically it kind of already gives you some information about how the variants might affect the person. So once we've done that, we're going to score our haplotypes, because typically, especially if we have several possible variants in close quarters, we might have multiple haplotypes, more than are possible biologically. Ultimately, we're usually looking for two. So we're going to need to score our haplotypes. Going back to the original reads that we had, now we have, coming out of the graph, we have a preliminary score based on how much support we had for nodes in the graph, but that's kind of a rough score just for the purpose of, if we have more than 128, which is kind of the current cutoff. If we have more than that, we only want to take the top, like the haplotypes. And that's just an optimization to avoid getting too bogged down in too many possibilities that are not real possibilities. But that's a kind of a rough scoring. Now we want to do a more precise scoring of our haplotypes. And so we're going to systematically realign every single read to every predicted possible haplotype and compute the likelihoods of those reads given the data. I'm not going to go into the mathematical details. Basically, it's using Bayesian statistics to be able to evaluate how likely the haplotypes are. And the only thing you need to really remember from this is that the key information that is used as part of the Bayesian modeling is the base quality. Remember yesterday we talked about base quality score recalibration, and we wanted to have very accurate estimations of how accurate the base calls coming out of the sequencer were. This is where we use them. So that's why it's important that they be accurate, because we're using them as part of the modeling process of how likely the reads are. It's basically a way of taking into account the quality of the bases when we're making decisions about the likelihood of the haplotypes and ultimately the alleles for the variants that we're calling. Okay. So take a look. Base qualities are important. This is where they come into play. All right. We're computing likelihoods for haplotypes, which may involve multiple different alleles at different positions. And so at this point, we have to go through a process that's called marginalization to be able to compute likelihoods specifically for each position. Again, more Bayesian math. The take home here is that we're looking at each position, how much evidence we have for each of the possible alleles, and that is going to allow us to make a decision about what is the most likely to be real in our sample. And that ultimately is what will allow us to make a decision about the genotype call and say, this person is heterozygous AC at this site. Okay. More details of the Bayesian math, and if you're interested, I'm happy to go over that at the break, but usually that's not something that everybody wants to know, and that's fine. The key thing is that you want to know from this part is that the modeling process takes ploidy into account. So by default, we're assuming that you have a diploid organism, and the model takes that into account right here, because we're looking at the two possible alleles that you might have within a diploid organism. But if you're working with something that is not a diploid, whether it's haploid or a fun polyploid with something exotic, that's totally okay. There's a parameter that you can give HaplotypeCaller to tell it, I have a tetraploid, please look at 4 possible alleles at this step. And so at this point, we'll just have more terms in the equation and take that into account. And that's how that works. So again, diploid by default, but if you want to use a different ploidy, you can set that. Chromosome-specific ploidy. I'm not sure that's currently possible. I think you would have to do separate runs of the program. Yeah. Yeah. Can you say that a little louder, please?
So if there are parallel situations,
Mm-hmm.
if you would increase the parameter and maybe to be logical peak probability improve the point of frustration. On other sides. On that.
Yeah. Are you thinking specifically of the sex chromosomes?
No, if there are such genes which is complicated between
yeah.
and that they are different between one base and then the bases are quite often misaligned.
Yeah, we don't have any recommendations for that, for dealing with that in general. We kind of assume that the mapper puts things in either one region or the other. We don't do any particular processing for that. That's interesting. That would be interesting to look at, but I don't have an answer for you.
But many of the clinical problems that we are dealing with,
yeah.
They are related to
That would be interesting to talk about to one of the developers who knows the model more deeply, to be honest. Yeah. Any more questions about this part? No? Am I speaking loudly enough? Because sometimes it goes back down after a little while. So don't hesitate to let me know. Okay. So at this point, we have values, we have numbers that we derive this way. And this is just in practice what it looks like if you were to write out the numbers involved. We're actually using numbers here that are unrealistically large. These likelihood normally would be much smaller numbers. And there would be a lot more of them because each one, we're looking at the support for each read. But so our example is not entirely realistic, but if you're interested in walking through the math, this is what it looks like. And what you end up with is a genotype probability for each of the possible genotypes that you're considering based on the alleles that you've selected. You're going to have a number that describes how confident we are that that is the right genotype for that person. We're going to do one more transformation, which involves normalizing that probability and putting it in Phred scale. So Phred scale is the same logarithmic-based scaling that we use for base quality scores. And in general, it's something that we often use to transform numbers that are difficult to work with and represent succinctly into numbers that are, most of the time we round up to integers because they take up less space and they allow us to capture the value without having to do as much work in our heads and in the file. So we're transforming, normalizing. We normalize so that the chosen genotype for that particular sample, in this case, the CC heterozygous variant SNP, the chosen genotype is always 0. It's just convention. And all the other genotypes are scaled so that basically they're, relatively speaking, they tell us the degree of confidence relative to the one that was chosen. And so the higher the value, the least likely it is that that is the right genotype based on the Phred scaling. And so if the values are very close together, and here you see these are very small numbers. Usually these numbers would be much larger, but it's because our example we were using with these unusually large probabilities for different possibilities. If the value between these is, if the difference between these is small, that means we're not very certain that we made the right decision. We may be very certain that we have a variance in this location, but we may not be sure whether the person is heterozygous or homozygous variant. And that's what's reflected here. And so your next most likely option is going to be used for the GQ, the genotype quality. The genotype quality is the value that tells you how confident we are that we picked the right genotype. Does that make sense? And these values are things that will come up. They're in the VCF. Those are the values that tell you whether you can trust the variant call or not. That's why I insist on them a little bit, walk through how they're derived. Because ultimately, whenever you have a variant call that's not super obvious, and you wonder, like, is this really the right thing? Those are the values you should look at to try to understand what happened and whether you can trust that in your downstream analysis or whether you should be more skeptical that that represents the biological reality of your sample. Sure. Yeah, so there's some base priors about the heterozygosity and things like that. And that is something that you can set. So if you have, and it's based on the expected heterozygosity in humans, but you can set that if you're working with a different organism or a population that has a higher mutation rate or something like that. That is, you can set that on the command line.
So it's the same prior for every side.
Yeah.
It's not based on the reference.
That's correct, yeah. So it's the same basic one, but I believe you can provide a list of priors if you want to give different priors for different regions, I think. I would have to check that. Any other questions? Okay. All right, to recap, we walk through, we look for active regions. Whenever we find a region, we go through the process of reassembling the reads with the graph, deriving possible haplotypes, scoring our haplotypes, and then finally assigning genotypes and working out the genotype qualities involved. That is what HaplotypeCaller does if you give it a single sample. If you give it a multiple samples at the same time, it will do the same thing, except like the graph assembly will use the reads from all the samples, which is powerful, but can very quickly become computationally difficult. So generally, we don't do direct multi-sample analysis. That's why we broke out the join calling into a separate part.
I have a question about, so that I'm not sure if I understand correctly, but so we have these spread scores at some point, and then we recalibrated them, and then at this point we have recalibrated spread scores for every genotype in the alignment, and now we are fine-tuning in some of the places where entropy is higher, and then the rest of them we keep it untouched.
Yeah, so, in the areas where there's no evidence that anything is variant, we just don't call variant.
And then we don't normalize any, and we don't do any normalizations for those spread scores that are untouched. And
what do you mean by Fred scores?
We have used some kind of normalized PL scores for those scores that are touched by this algorithm.
Yeah, so we can calculate those PLs for the variant sites. Okay. We do actually, I didn't go into it, we have a similar calculation for the non-variant sites. Okay. And we'll talk a little bit more in the next bit about that. It's basically the same system where we spent a lot of energy looking at variant sites to provide statistics to accompany our variant call. But we do a similar, though lighter process for non-variant sites, so that for each non-variant site, we can have an estimate of how confident we are that it's actually non-variant versus just not having enough data. And we'll go over that in the next talk. And that's basically what this is saying. So we did that per sample. In GVCF mode, we emit that extra information about the non-variant sites. And so in this next step, we're going to be able to take that information and do a join calling, looking at the variant calling information from all our samples and decide across our population what are real variants versus not. These are just the command lines for reference, but you'll actually run them in practice in the practical. And so the next step is going to be joint calling. Any more question about HaplotypeCaller?
So about the PL and the TQ. So how can I know what is a good number or not?
It's relative. The GQ, so the genotype quality, is the one that will usually give you a really good sense. Most sites will end up with a GQ in the 90s, somewhere between 90 and 99. Technically, you can have a higher score, but we cap it at 99 because anything above 99, we're super sure, and there's no point in knowing that it's 200. 299 means the same thing. Super sure of this genotype. Below that, there's kind of it's kind of the range. Anytime, it's hard to give you exact numbers, but I would say as long as you're in the upper, it's kind of like temperatures in Fahrenheit. As long as you're above 70, you're usually pretty comfortable. Anytime it drops below that, you start being more skeptical. And that's when you look a little bit more deeply at what's happening, looking at the statistics, if that makes sense. Just think about how comfortable you are in a Fahrenheit scale of temperature. All right. So that completes it for HaplotypeCaller. And our next talk is going to be Andre talking about joint calling. And that's going to take us to coffee break. So one more talk before coffee break. Survive that long, and you'll have coffee, and then we'll do practicals until lunch.